Thank you, Andrew, for the introduction, and thank you all for listening. I'm excited that you're interested in this topic, and I'm excited to share some of my research with you. This was research that I did as part of my dissertation at the University of Pittsburgh. And can't advance the slides, which is a problem. Okay, so today, first I'm just going to walk through a broad introduction of the pollen virome and what I mean by possible ecological drivers of it. So to begin, ecological drivers are just things that might influence the pollen virome of a plant species. Then I'll talk about the methods that my team and I used to answer our main questions. And our main questions for the project that I'm going to talk about today were what viruses are associated with the pollen of wild plant species and what is their host range? Number 2, do plant traits influence the pollen virome? And number 3, do landscape traits influence the pollen virome? Lastly, I'll summarize our main findings and how they fill in some of the gaps of our knowledge about the pollen virome, especially of wild plant species. So first, the introduction. So I think as we've all become very aware in the last couple of years, viruses are ubiquitous. They infect nearly all life forms across the animal kingdom, the plant kingdom, and the microbial kingdom. But what is the virome and how much do we really know about it? So we can define the virome in general as all the viruses associated with an organism. It's hypothesized that there are 1 × 10⁻31st power viruses on Earth, and most of these are totally undescribed and probably unrelated to those that we already know about. Most of the ones that we do know about, though, are important to our livelihoods because they cause diseases like the flu, AIDS, and COVID, all of which are very serious and make people very sick. But what if we zoom in a lot and think only about the plant virome, which are all viruses that are associated with a plant? There are about 3,000 described plant viruses. Many of these are vectored by herbivorous insects like beetles or aphids. And similar to viruses that cause human disease, focus has mainly been on plant viruses that directly impact our lives or cause damage to or yield loss in plant species of agricultural or economic importance. And then finally, if we zoom in even farther to one specific part of the plant, what is the pollen virome? So the pollen virome is a subset of plant viruses that are associated with pollen. So this means that they could be on the outside or the inside of pollen grains. And pollen-associated viruses are vectored by pollinators, like bees, as they forage for floral resources like nectar and pollen or other things that they might use to build nests. There are approximately 100 viruses and viroids that have been described as pollen-associated so far, and most of these have also been studied in agricultural or economic contexts, just like the herbivore vectored plant viruses. So though pollen is a pretty unique viral niche, most of what we know about it is limited to how pollen-associated viruses spread between and infect susceptible individuals. So for instance, we know that pollinators such as bees are vectors. Plants can't walk around and infect one another like we can. So pollinators are necessary for pollen-associated virus transmission. Pollinators transfer pollen from the anthers of flowers to the stigmas of flowers as they forage for floral resources. If the pollen is infected, the pollinators are also transferring viruses as well. And the stigmas are really highly vulnerable and receptive surfaces. So you can imagine that viruses might easily enter into that area. And pollinators can also wound reproductive tissue while foraging for floral resources, or bring infected pollen into contact with a fresh wound on a plant. And I'll talk a little bit more about all of these pathways in the next few slides. So from past research, we also know about how pollen-associated viruses infect susceptible individuals, not just how they move between individuals. So pollen-associated viruses can infect vertically. So this means that viruses infect susceptible offspring via a parent. In the case of pollen-associated viruses, this does not go from the mother to the offspring. This goes from the father to the offspring. And once the pollen is transferred onto the stigma, that pollen can germinate. The pollen tube that then comes from a germinating pollen grain grows down the style and delivers the sperm housed within the pollen grain to the egg that is within the ovule of that receptive flower. If the pollen is infected and the fertilization of the egg by that infected sperm from the infected pollen grain is successful, because it isn't always, then the offspring might be infected as well. Past research has also focused on horizontal infection. So this means that pollen-associated viruses can infect susceptible individuals horizontally, meaning they move from an infected individual to a susceptible individual. This can occur after pollination in a number of ways. In one way, a pollen-associated virus could move into the tissue of the stigma or a style when the pollen grain is germinating or that pollen tube is growing. Or after pollination and after fertilization, a pollen-associated virus can move from a developing embryo to the mother through maternally derived tissues that connect that embryo to the mother if the fertilization event was successful. Lastly, horizontal infection can occur after a pollen-associated virus penetrates a fresh wound in either reproductive or vegetative tissues. I think this is really interesting because some pollen-associated viruses can infect susceptible individuals both vertically and horizontally, meaning that they are capable of infecting more than one generation of a plant in a single infection event. So we know quite a bit about how infection by pollen-associated viruses occurs and how they're transmitted. And that information is, of course, really important. We know far less about the ecological drivers that may influence the pollen virome. These could include things like virus traits, plant traits, pollinator traits, and even traits of the landscapes in which the plants are growing. So you can see that there are nearly endless things one could ask questions about and see that all of these things are interconnected in some way. Today, I'm going to focus just on a couple of plant and landscape traits. One of the many possible ecological drivers of the pollen virome are plant traits. More specifically, these can include flower traits that mediate interactions with pollinator vectors and pollen grain traits that make pollen collectability and transfer happen at a greater rate. For instance, having multiple flowers is one trait that increases the number of interactions between a plant and its pollinators. Having a more closed flower morphology or shape is a trait that leads to more intimate interactions with pollinators. Higher intimacy in plant-pollinator interactions increases the likelihood that pollen and then perhaps pollen-associated viruses will be transferred to a stigma. And lastly, spiky pollen grain exines, or the outer layer of pollen grains, help pollen cling better to pollinators, which can also increase the likelihood of transfer of pollen grains and maybe those pollen-associated viruses to recipient flowers. And these are not all of the plant traits or flower traits that could be important here, just a small snapshot. Another of the many possible ecological drivers of the pollen virome are landscape traits. Some landscapes are heavily fragmented by human land use, while others are relatively isolated and experience very little or no human land use and are therefore dominated by natural vegetation. Others lie somewhere in the middle of this continuum. One example of human land use is agriculture. Viruses in general often emerge and thrive in agricultural environments because potential plant hosts are packed so closely together. And pollen-associated viruses specifically can also emerge and thrive in these environments, since crop species and wild plant species may share pollinators, and pollen may be transferred between the two groups of plants. So since past research has focused on pollen-associated virus transmission, vertical infection, and horizontal infection, we have quite a lot of mechanistic knowledge on how pollen-associated viruses infect susceptible individuals. But we don't really know the ecological factors that influence the pollen virome. So in other words, we're currently lacking the big picture of the pollen virome, especially in wild plants, which is why we wanted to ask, again, what viruses are associated with the pollen of wild plant species and what is their host range? Do plant traits influence the pollen virome? And do landscape traits influence the pollen virome? To answer those major questions, my team and I used a variety of field, lab, and computational methods. For the main project that I'm going to show data from today, we collected pollen from 24 plant species in 4 different landscapes around the United States. These plant species were all herbaceous and animal pollinated, and most were perennials. The plant species were also all wild and not species of agricultural or horticultural interest, because one of the biggest gaps in knowledge concerning pollen-associated viruses is what is going on in the pollen viromes of wild plant species. And the landscape trait that we focused on was the level of human land use. Specifically, we focused on the amount of impervious surface and agriculture together as the metric of human land use. The landscape that had the highest amount of human land use was what we were calling the Eastern Deciduous Agroforest Interface. It's heavily fragmented by agriculture, and it's characterized by 56% human land use. This GIS picture here shows, and others that I will also show on this slide, show impervious surface in red, agriculture is in yellow, and natural vegetation is in green. The California coast landscape is characterized by dunes, scrubby vegetation, and some urbanization, and characterized overall by 12% human land use. The Central Appalachian landscape is characterized by mostly preserved habitat in the Blue Ridge Mountains and has about 11% human land use. And our last landscape, the California grasslands, is dominated by very remote areas of native chaparral vegetation and serpentine seeps, and it has the least amount of human land use at only 2%. The plant species in this project differed in several floral and pollen grain traits, which we chose to focus on because they're important for interactions with pollinator vectors or pollen grain collectability, and therefore we thought they might be the biggest drivers of the pollen virome. That's not to say that there aren't others, because there probably are. The traits included flower size, the inflorescence type, so whether a plant has just a single flower or multiple flowers presented at a time, longevity, or for how long a flower lives, reward type, and so whether a plant has nectar or pollen or both, floral symmetry and reward accessibility, and pollen grain texture, and pollen grain size. We hypothesized that versions of these traits that increase plant pollinator interactions, like having multiple flowers instead of a single flower, or more open flowers instead of more closed morphologies, or having spiky instead of smooth pollen grains, would lead to a plant species having more pollen-associated viruses. In the field, we collected a lot of flowers, and then we sonicated the anthers so that we collected only the pollen. I want to stress that this was done in as sterile a manner as possible in the field, and we were careful to remove any plant tissue or anything else that found its way into the pollen before we closed the tube and put it in the cooler. Then back in the lab, our first step was to freeze dry the pollen, because pollen is a very tough, resilient plant product and would not break otherwise. So then after freeze drying the pollen, we did use a machine to break the pollen apart, and we checked to make sure that about 50% of the pollen grains in the sample broke. And then we extracted the total RNA from the pollen grain samples. And we focused on broken up and whole pollen grains because we wanted to capture the viruses both on the inside and the outside of the viruses. And we extracted RNA instead of DNA because most plant viruses have genomes that are made up of RNA. After getting all of the total RNA out of the samples, we sequenced one sample for each plant species, and then we used a couple other computational methods, including a viral discovery pipeline, to identify the known and the novel viruses associated with each of our pollen samples. And in the next few slides, I'm going to break down what I mean by the known and novel viruses. We used a computational pipeline to identify the known viruses and discover new ones in all of our pollen samples. So first in the pipeline, there are some quality control steps and a step where the host plant genome is subtracted away from the sequences that we had. What was left were the pollen-associated virus reads or non-plant reads. Second, we assembled those reads into longer sequences called contigs, and we aligned both just the reads and also the longer sequence contigs to different protein and nucleotide databases containing viral reference genomes. And based on how well our contigs and reads matched, we were able to say what was a known virus and what was a novel virus. We identified the known viruses when aligning the reads to the databases. The reads had to align to a reference genome in a database at least 10 times and cover at least 20% of that reference genome. We had these constraints because we wanted to make sure that we were identifying viruses that were truly present in or on the pollen grains and that were biologically meaningful and not some sort of artifacts or environmental contamination of some kind. We then identified novel virus or discovered novel viruses through contig alignments to the same protein and nucleotide databases. We called a contig a novel virus or a virus that has never been described before if a contig was less similar to a reference genome than the species demarcation criteria defined by the International Committee on the Taxonomy of Viruses. We then broke this novel virus category into 4 finer scale groups. One of the best groups was this idea of a novel coding complete viral genome. This included contigs that had sequence lengths that were long enough, and we were able to detect enough of the proteins belonging to the virus family that we thought that a novel virus belonged to. Very similarly, we also were able to identify discover novel coding complete strains of known viruses. So this is essentially the same as the first category, with one important difference. These contigs were a better match to those reference genomes than the totally novel viruses. And it was interesting during this whole process, just learning about viruses in general and trying to figure out how the ICTV classifies viruses, and these species demarcation differences are different for every family. So it was really interesting to think about why it might be different for everyone. But then the other two groups of novel viruses were these novel partial viral genomes and novel partial strains of known viruses. Again, these follow kind of the same criteria as the slide before. However, these were sequences that were much too short for us to say if with high levels of confidence that they belonged to a virus family. But we did take notice when we saw something called an RNA-dependent RNA polymerase, or an RDRP region, because these are highly virus specific and are really useful in RNA virus classification. So I wanted to go through all of that kind of technical detail because it was really important in how we actually quantified the pollen virome. So when I present our results, I will refer broadly to this idea of pollen-associated virus richness, which is the total number of unique viruses that we found to be pollen-associated for a given plant species. A more conservative estimate of the pollen virome, or the pollen-associated virus richness, was the total number of those known viruses, the total number of the novel coding complete viral genomes, and the novel coding complete strains of known viruses. So all of the things that we were very confident were actually in those pollen samples. We had a little bit of a relaxed estimate of pollen-associated virus richness as well, which included everything from the conservative estimate, but also the counts of those RDRPs of the partial viral genomes as well. So as a reminder, our first main question was, what viruses are associated with the pollen of wild plant species, and what is their host range? So to illustrate the answer to this first question, I'm using this presence absence grid, and the plant species from the project are across the top. The viruses that we found or discovered are across the side, and the blue squares in the middle correspond to what region or what landscape the plants and the viruses were from. We found 22 known viruses, and interestingly, not all of them were plant viruses. One was a virus that infects bees, the deformed wing virus. 10 of those known viruses were found in more than one plant or landscape, and they're shown there with the purple stars. And we also discovered 6 novel coding complete viral genomes and 3 novel coding complete strains of known viruses, which are underlined in orange. Our second major question was, do plant traits influence the pollen virome? After getting a sense of what the pollen virome itself looked like, we performed what's called a principal component analysis of the floral and pollen grain traits that I mentioned before. The dots in this figure represent each different plant species, and I am just showing this figure to you to show that the plants did vary quite a lot in those 8 traits, which is why the dots are spread all over the figure. And the most important traits for PC1 were the number of flowers in an inflorescence and the flower symmetry and reward accessibility. The most important traits on PC2 are the pollen grain texture and size. We then use these PCs along with another one from this analysis as predictors in a linear model to actually be able to say whether or not these traits influence the richness of the pollen virome of the 24 plant species in this project. So this graph is showing the values of PC1 along the x-axis and the log transformed conservative estimate of virus richness along the y-axis. Again, the colored dots represent each plant species, and those dotted lines are just the 95% confidence intervals. Somewhat contrary to our initial hypotheses, we found that plant species with multiple flowers, bilateral symmetry, and restricted access to floral rewards had more pollen-associated viruses. This graph is the same layout as the one on the previous slide, but this time the traits that are important to PC2, so those about the pollen grains, are along the x-axis. Also somewhat contrary to our initial hypotheses, we found that plant species with smaller, spiky pollen grains had more pollen-associated viruses. Our last major question was, do landscape traits influence the pollen virome? As a reminder, in this project, the 4 landscapes varied quite a lot in the amount of human land use, which was our landscape trait of interest that we thought might influence the pollen virome. In this figure, the stacked bars next to each block of plant species from the 4 different regions show natural vegetation in green, impervious surface in orange, and then the gold color is agriculture. And just to show again the variation, the California grasslands region had only 2% of human land use, and the Eastern deciduous agroforest interface had 56% human land use. Here I'm showing the landscapes along the x-axis, and again, that conservative richness, or the conservative estimate of virus richness along the y-axis. And we found that plant species growing in that Eastern deciduous agroforest interface, or the landscape with the highest amount of human land use, had the richest pollen viromes. In contrast, the plants growing in the California grasslands, or the place with the least amount of human land use, had the least number of pollen-associated viruses. Now that I answered all of our questions, I want to summarize the main findings and think about where this work could go from where it is right now. So in this project, we did identify many viruses, and all the viruses that were identified or discovered, 9 virus families were represented, and more even if you include all of the little partial pieces that we found. This indicates that pollen-associated viruses are very prevalent and very diverse in wild plant species, and some of them also seem to have wide host ranges, since some of those known viruses were found in more than one plant species or even more than one landscape. If this is in fact true in other systems and for other plant species, pollen-associated viruses, if they're actually harming the plant, could threaten wild plant biodiversity, especially if the pollen-associated viruses originate in a crop plant species, for example. We showed for the first time that plant traits do influence the pollen virome. So as I had mentioned before, a lot of the work on pollen-associated viruses is pretty constrained to one focal plant species and one virus, and elucidating the mechanisms behind the infection. Here we're showing pretty broad patterns that plant species with flower traits that mediate increased intimate interactions with pollinators and pollen grain traits that are important for pollen grain collectability do influence the pollen virome and lead to plants having a richer pollen virome. Lastly, we also showed that landscape traits also influence the pollen virome, as we found that plant species growing in landscapes with higher amounts of human land use have more rich pollen viromes. So though this project was really exciting, there are still many unexplored ecological drivers of the pollen virome. So to get a good sense for what is really going on, I think first more observational studies like this one need to be done, and then perhaps more studies involving the transmission, either on pollinators or just more mechanistic studies would be really useful. But all of these ecological drivers are connected and could include things like pollinator traits and their foraging behavior, other landscape traits, not just the level of human land use, but perhaps the level of invasive plant species in a landscape, or overall plant density. Could also include virus traits, like the type of proteins a virus has, or whether it can infect plants vertically or horizontally, and also many other plant traits related to pollen grain architecture or chemistry or just lifespan of the plant in general, along with many, many others, could be important to really understand what's going on in the pollen virome of wild plants and understand what drives its diversity. So I think I'm ending a little bit short, and I apologize for that, but I do want to say this work would not have been possible without all of the co-authors that worked with me on this project, along with all of my dissertation committee members and people that were in the Ashman Lab at the University of Pittsburgh, both past and present, along with the funding sources and the facilities and field sites that we were allowed to use. So with that, I'm happy to take any questions that you might have and just brainstorm too if you want. And thank you.
Thank you. That was super interesting. We do have a couple questions in the chat we can start with, and if folks have questions they'd like to ask too, you can raise your hand in Zoom and we can just call on you to ask. Start with Sharon asked, are all viruses pathogenic?
No, they are not. So that's a great question. And for our project, we couldn't tell what the viruses were doing to the plants. In fact, in the field, when we collected pollen, we tried to get pollen from plants that didn't look like they were sick at all. So the viruses that we saw, or that, like, they may not have been harming the plant at all. Infection doesn't mean harm, and we have no way of telling in our project what was going on.
Interesting. Elena, you touched on this a little bit, but has there been any more research looking for viruses that use pollen or nectar as a vector to reach pollinators and bees?
Um,
You mentioned you found that
oh, yes.
the
Sorry.
bee wing virus. Yeah,
Yeah, sorry. It took me a minute to really understand
I
the
didn't
question.
phrase it the best way.
No, it's okay. So there is a large body of research that shows that some bee viruses, like deformed wing virus, and I'm now blanking on some of the other big viruses that infect honeybees, but they do travel on either contaminated pollen or on the flower surface. So if an infected bee lands on that flower and goes to the restroom, another bee can come by and pick up that virus or even bacteria, because some of them work that way too, and take it back to their colonies if they're honeybees or bumblebees, or if they're solitary bees. Like, they can also pick up viruses and other diseases that way as well.
And Sharon asked, were the viromes in Appalachia the same as the viromes in California?
Um, Not exactly the same, but there were some overlaps, and I can go back somewhat quickly, maybe, to the one presence absence grid. So the like medium blue color and the super light blue color which looks pretty washed out on this diagram, we're both in the California area, and then the Central Appalachian region, where it's kind of this more like sky blue area. And I don't think there were a lot of overlap, if at all, with those two regions, which is interesting.
Yeah. Ronnie asked, in the context of evolution, what are some of the effects of the viruses on plants, and does that help the viruses be successful? How does that help the viruses be successful? Sorry. What are
Okay.
the evolutionary pressure on the plants from the viruses? Yes.
I'm trying to think. That's a good question, and I don't know if I know the answers to that one. If Sorry, let me think about this for a second.
They said that a lot of the viruses aren't pathogenic, so maybe
Right,
a lot of them might not really have any selective pressure on the plants, but
right.
if there are some that do, maybe favors those traits.
Yeah, some that do. So, for example, and I'm going to think about this from actually the virus standpoint instead of the plant standpoint. So viruses that are able to infect plants horizontally usually cause more problems for the plant. There are a lot of viruses that just hang out in the gametes of the plant, and those are the ones that are only transmitted vertically. They don't cause any problems, they're just there. But for the ones that can go either horizontally and vertically, or maybe just horizontally, that's when you do see things like, like the phenotypic characteristics of a viral infection, like ring spots on a leaf, or damaged fruit, or other, similar characteristics. And so if the virus is actively infecting the host, there are ways that the plant could potentially respond and shut down that viral infection, but I am not sure about the like time that it takes that to occur and how long over evolution that occurs. I'm So sorry, that's a terrible answer to the question.
Some new field of study, sounds like. Well,
Yes.
Connie says thank you. Uh, Stephen asked, are there more pathogenic viruses associated with areas utilizing domestic honeybee pollination services, for example, almond orchards?
Possibly. So there is a worry that there is some viral spillover, not just potentially between plants, but between domestic honeybees or bumblebees into solitary native bee populations. And I don't think we know yet how much that's affecting the solitary bee populations and communities, but it is a concern for sure.
Um, Tarn had a question. Just it was a little hard to read the
Mhm.
plant species on this chart.
Uh-huh.
If you were asking if you looked at the same species or genus in the four different areas, and if you found similar viromes across the same genus or species.
So we did not. For this project, we some of the families, the plant families that were represented in each of the like blocks of plant species from the regions, those were um the same in some areas, but we did not have any overlap with species at all, which is a really interesting question and something that we actually thought we would do when we started this project, but it's very takes a very long time to collect at least 10 milligrams of pollen, and some flowers are it just doesn't work out, and there's not enough of those flowers in an area to do that. So we dropped any kind of paired study or paired design very, very quickly from this project, which is unfortunate. But we also think it would be really cool to look not just at the same, plant species in different regions, but also maybe even the same plant species, if this was possible, in areas where there's, you know, not a lot of human activity with maybe some human activity and then maybe a lot, just to get a sense of like truly how much is the landscape driving what we're seeing, or is it the plant themselves? Like we're not sure. And this was um, very broad survey project, really, more than anything.
Yeah. Beth asked, what is the current thinking or research on effects of climate change on these viruses in wild plants? Is there any specifics you can share about that?
Um, I'm not sure about climate change per se. However, as we are continuing to alter landscapes and we are continuing to grow larger and larger monocultures of crops, it is a concern that viruses can move between crops and wild plants or vice versa and cause issues either for the crops or the wild plants. So I know that's not exactly about climate change, but I think it's definitely related.
Mhm. Yeah, land use and climate
Yeah.
change questions are closely linked.
Mhm.
So when you're collecting RNA from the pollen, how do you separate the viral RNA from RNA that would normally be expressed in a pollen?
Yes. So we the beginning steps of the pipeline that our collaborating lab designed to detect viruses and pretty much anything, which is really cool. It subtracts out the host genome. So in some cases, we couldn't get like an exact match between our host plant species and whatever was in like the NCBI genome database, because they just simply haven't been sequenced yet. But in most cases, we could get close enough where things that came out, like the output of the pipeline, were, yes, we had things that weren't just viral. There was fungal, there was bacterial, there was a box that nobody, a sheet that nobody knows what it is because it didn't really match to anything. So we focused just on the things that matched closely enough to viral reference genomes, and move forward with that. But there's, again, a whole bunch of data we didn't even look at from this output. But that's basically how we could tell.
Related to that, what is the significance of the RNA dependent RNA, Paul?
Yeah, so that is what how the RNA viruses transcribe their genes. And so they are extremely virus specific or species specific. And so when you see that in a sample and it, isn't matching to anything else, you're like, okay, so this virus was present, especially if we could see like the whole region bioinformatically. Again, we didn't do any of this like on bench work or anything like that, which would be like another avenue for research. But it's just that it's very useful and in classification studies, and it's kind of like the benchmark that is used for at least plant viruses. You can also use things like coat proteins, or is the other big one, but we chose the RDRP.
Yeah, I told Andrew before we started, but a lot of you know I study bioinformatics, so I
Yeah.
have a little insight into this too.
Yeah, so hopefully you enjoyed my very quick overview of the, like, how did we discover this?
Yes, yeah, it's good. What was the like ratio of known to novel viruses that you found?
Way more novel viruses, which is kind of in line with the hypothesis that there are so many viruses out there that we have not even begun to scratch the surface with what we've described and what we haven't. So I thought that was pretty cool to me, to be like, wow, like look at all these pieces of viruses or full viruses that, nobody knows what they are. And I think it would be an interesting avenue of research to take some of these novel viruses and learn about how they infect their host plants, like kind of go back to some of those like more mechanistic questions and ask, like, do these do the same thing as other plant viruses in this family that they belong to, or do they do something different because they're more pollen specific? Like, nobody knows the answers to those questions.
Yeah. Well, building on that, like, what do you think are the most interesting or relevant like next research questions and or experiments to do in this field or in this topic?
I think learning more about how these traits, and I say these traits, I mean like traits from either plants or pollinators or both, connect with each other to influence the pollen virome. We had started a project that we will probably publish in maybe a shorter paper than what we originally thought. We wanted to try to show that like plants that share pollinators also share viruses. And while we maybe found hints of that, it's really hard to say that, those viruses arrived at the plants on the pollinators, which we're pretty sure they did. But again, with the project design, we can't fully support that. So it's kind of hard to say where they came from with 100% certainty. But I think, not just the landscape traits that drive the diversity, or the traits of just the plant or just the pollinator, just the virus themselves, but kind of the interplay between them too is also really, really important because all of these things interact with one another.
Yeah.
So again, that's more like observation and kind of like pattern finding, but I think that's important, especially as we do continue to alter landscapes. Like, I think we need to get a kind of baseline understanding of what is happening and then see maybe what could happen. And then see like more directed studies of like what are the viruses doing in these plants? Are they hurting the plant at all, or are they just there?
Yeah, Matt, Sharon put a question in the chat related to that. Is it fair to say that the viruses are doing more harm to the pollinating bees than to the plants?
So these viruses, it's hard to say. I don't, so some of the some of the known viruses that we found, and I'm just reminded of this because I glanced at this presence absence diagram again, like they do cause problems for specifically like crop plant species. They damage the yield and also not sure if these pollen-associated viruses are also transmitted by herbivores. That's also another very interesting question that nobody knows the answer to either. So I think the short answer here is we don't know. The ones that are harming bees, that are bee specific, yes, they are definitely harming the bees more than they're harming the plants, like the deformed wing virus and any others that might travel this way. It was also really interesting. There was a paper that came out a couple of years ago that showed that a plant virus was actively replicating in honeybee tissue, and it wasn't replicating in like their salivary gland or their guts. So it wasn't just like, oh, the bee ate or some pollen or some plant material with the virus in it. Like it was replicating in other tissues in the bee body, which that's all they showed. They didn't make any kind of, conjecture about like what that virus is actually doing. But I think it's really, really interesting that somebody has shown that, because that also says that, okay, maybe these viruses are jumping hosts, and not just like jumping plant hosts or pollinator hosts, but like crossing kingdoms, which is crazy.
Yeah. Sally asked, even with all the novel viruses, are there still fewer than 100 known associated with pollen?
So if you take like all the viruses that we found, I guess the answer to that is no, there's not less than 100. However, what there's a couple of steps that you have to go through to show that a virus is in fact only pollen associated, and it is in fact only the pollinator vector that brings that virus to the plant. And we definitely didn't do that. We just found some viruses. And so again, somebody could build an entire like research program based on this and take some of these viruses that haven't gone through all these like rigorous experiments and ask like what how did they get here? Are they pollen associated? Are they truly novel? What are they doing? I think that would be interesting.
Absolutely. I think we got through all the questions, unless anyone has any final ones. But I want to thank you again, Andrea, for joining us. As a really interesting presentation. Definitely a topic that I didn't know a lot about. So it's really helpful, and hopefully see everybody next month for our last presentation, and have a great rest of your evening.
Yeah, thank you, everyone. And thanks, Andrew.
Night.
Good night.